All right. Welcome to the A's Philosophy YouTube channel. I am A's Philosophy, and today we are going to be doing our third installment of the A's Philosophy. Excuse me, just doing a sound check there. Of the ID update, and I think today is going to be a big one. I think today is going to be really important for people who are interested in this topic, and I don't know why anyone wouldn't be interested in this topic. I think that uh, the question of whether evolution is true is extremely important, mainly because a lot of people are being taught that it's true and believe it's true, but there is some extremely strong evidence that it's not true, and it's based in science and empirical fact, essentially. And today we are going to be discussing a topic that it's hard. It can be hard to understand at first, but once you do understand it and once you grasp it and realize its implications, I think that one can see that you can make the claim that these scientific observations falsify evolution. And the topic is, as we see in the title screen here, developmental gene regulatory networks, which to the average person, I think might look at that and say that's really boring, it's really complex, it's difficult to understand, and what does it really prove in the end? And that's reasonable. I'm just hoping people will give this a chance for those who haven't already looked into this, which is probably most people, because we really are seeing the collapse of the evolutionary paradigm. In my opinion, evolution is a completely dead and false theory. And there are so many observations in science that prove this and demonstrate this. I think evolution is hanging on by a thread. It's hanging on by public perception. It's hanging on by the fact that it's been believed and taught by the scientific community for so long. But there are some things under the radar that just the public is not aware of, such as this topic, that provide strong evidence that there's reason to question these narratives. There's reason to question these paradigms. So yes, developmental gene regulatory networks. First of all, what we're going to do, we're going to discuss what they are, and then we're going to look at some articles written once again on a couple of them on the Evolution News and Views website, which is an outfit of the Discovery Institute, talking about the scientific research regarding developmental gene regulatory networks and why they pose such a tremendous problem for evolutionary theory, why they really do refute the predictions being brought forward by evolutionary biologists. And make no mistake, the evolutionary biologists are aware of this. Of course they are. They're the ones who study these things, and they have their answers, and we can take a look at those things. But first, we're going to look at we're going to try and get an understanding of these things. What are they at a basic level, at a conceptual level, and then explain why scientific experiments are showing that they provide these problems. So let's go ahead with that. Let's see if we can get this moving real fast. All right, this is a big block of text. Again, I'm going to try and break everything down as simple a level as possible. First, we have to go through the process of actually giving the scientific explanation, so that that's out there and everything is being put in context, and then a fuller explanation and simplification can be brought forward. So what are developmental gene regulatory networks? All right, so this is going to be difficult. It's going to be we're going to be moving our way through this, but when we get to the end, we're going to have a better understanding of things, and we're going to see why problems are arising for evolution. Developmental biology. This is actually a quote from a book written in 2000 or published in 2013 called Darwin's Doubt. It's an excerpt from the book, but I think it provides a good definition of what developmental gene regulatory networks are. So this is the basic definition of these things. Developmental biologists have discovered that many gene products, proteins, and RNAs needed for the development of specific animal body plans transmit signals that influence the way individual cells develop and differentiate themselves. Additionally, these signals affect how cells are organized and interact with each other during embryological development. These signaling molecules influence each other to form circuits or networks of coordinated interaction, much like integrated circuits on a circuit board. For example, exactly when a signaling molecule gets transmitted often depends on when a signal from another molecule is received, which in turn affects the transition of still others, all of which are coordinated and integrated to perform specific time-critical functions. The coordinated and integration of these signaling molecules in cells ensures the proper differentiation and organization of distinct cell types during the development of an animal body plan. So that's a lot of scientific jargon. Some people may be able to easily understand it, some may not. But let's just try to break this down. Basically, when you have an animal that is developing as an embryo, obviously starting off as a single cell, the zygote, the sex cells combine to form the first cell of the organism, which begins the process of embryological development. You can imagine that that entire process you don't go from a single cell to a fully developed embryo randomly. There has to be control systems. It has to happen in a logical order, in a flow pattern. So it turns out that there are gene products, namely, as we see here, proteins and RNAs, which are produced early on in embryo, which basically control the entire process from beginning to end. And they activate at specific times and in specific places where they are necessary and when they are necessary. So you can imagine some body parts might develop earlier than others or at different rates and obviously at different places within the location of the animal's body. All of this is being controlled in a very specific way by these gene regulatory networks. Genes are activating in different places, different times to essentially regulate the embryological developmental process. All right, this is really, really key to understand. It's simple, really, when you think about it, but the implications go way beyond the simplicity itself. Like I said, the entire process is strictly coordinated. It couldn't happen any other way. So the coordination happens by the activation of various genes and the products at specific time, the products of these genes at specific times. And when you think about how complex that is, even for a simple organism, even some of the simplest single-celled organisms, this is an extremely complex process. There are a number of genes that are involved in this, and they all have to activate and do what they do basically at a very specific time in the process, or everything or nothing works. And we'll get to that. But basically, this is the product. It's just the idea that there are genes in the genome which control the development of the organism as an embryo. Okay, it's really not all that complicated when you think about it. It's pretty straightforward. And again, that entire is from Darwin's Doubt, 2013, pages 264 to 265. Okay. So just to get a diagram of what exactly these look like from a schematic viewpoint, we can see here, this is actually the developmental gene regulatory network for a sea urchin. And this was developed after many years of research by scientists who were studying, basically they would knock out certain genes at certain times within the organism and figure out what issues that created within the organism in the developmental process. And unfortunately, it led to a lot of dead sea urchins or sea urchins with gross deformations, which is sad. But they were able over time to develop this chart of how this organism develops as from a single cell to its fully grown state. And as we can see here, I'm not going to obviously not going to display all the details, but it looks like a computer circuit board. You see up here it starts, and there's a cascading effect, and the things at the top signal at certain times, and then they affect things downstream, and everything proceeds from there. And a sea urchin, while you could say is a very complex, but it's far from the most complex organism. But it's far from the simplest organism. But we can see here, I think this is extremely interesting to see how precise and exactly coordinated all this is, and all the parts that have to be in place for the sea urchin to get from a single cell to a fully grown, fully developed embryo. It's pretty amazing, and it all happens in a very specific pattern in a very specific way. And I think just thinking about how molecules floating around in space figured all this out. Remember, it has to basically, and we're going to get into this, it all has to happen the way it does. Okay, if anything's out of order or doesn't exist here, the sea urchin can't form. The heart, I don't know if sea urchins have, but basically the important bodily organs of the sea urchin are controlled by these developmental pathways. And if anything in this chart is missing, then especially in the beginning, but also towards the end, then this very serious parts of the organism don't exist, and the organism can't survive. So it all has to be like this, and it all has to happen in this way, in this very coordinated fashion. It looks like something a human or an intelligent being would create for a computer program, and that's essentially what dGRN's. They're computer programs as by way of analogy, of course. They're essentially software programs in the DNA of an organism that program how it develops during development. And thinking about how all of this unfolded in evolutionary history is mind-boggling. That mindless molecules just somehow over time just mutated themselves to figure out all these pathways. So even without looking into all the problems we're going to see going forward, it's still kind of raised a lot of questions about how this could have happened in the first place, even without the very serious issues we see going forward. How did, it took scientists years and years of study just to figure out what these pathways are. And they had to do it by basically killing the sea urchins and knocking out the genes that do these things, which leads you to the bare observation that, if these pathways aren't there, then the organism can't exist. So this is a serious, just the complexity of this is really astonishing. That mindless molecules, remember, the molecules don't care if they're in the shape of a sea urchin or if they're in the shape of a clod of dirt or a rock. They really don't care. But somehow they managed to create these extremely complicated circuit boards that develop the development of that control the development of an organism from one cell to many cells. It really is fascinating to think that this just happened just from blind chance, essentially, just like molecules blindly moving around in space, which is all the atheist really has. And not just the atheist, anyone who believes in the materialist narrative has to believe that blind atoms moving around in space figured all this out to create a sea urchin. So it's not just a complexity, though. There are serious problems that arise when you start studying these things as it relates to evolutionary biology. And we're going to try and get into as much of that as we can. So basically, let's see here. All right, so that is that. There's an article. It's a very long article. But we're going to try. We're not going to read all of it just because for time's sake, it's just too much. So I'm going to try and hit the highlights. This is an article. Basically, there's a field in evolutionary biology called evo-devo. Excuse me. And we're going to look into this. It's basically a different idea, a take on evolution that says basically, and this makes a lot of sense, for animals to evolve, these developmental pathways had to evolve because the developmental pathways are what take an organism from a single cell to whatever the final product is. So if you don't mutate the pathways, then you're not going to get to another organism. The developmental pathways have to evolve themselves. The developmental pathways are what are creating the organism essentially. It's not just the proteins and the genes for eye color and texture, fur texture, and things like that. The developmental pathways also have to change in a very coordinated way. And scientists recognize this, so they've come up with this idea called evo-devo, which says that not only do you have to change, I guess it all kind of works together, but basically the developmental pathways of the organism must change along with the other features of the organism in a coordinated fashion. And this field of study within evolutionary biology is called evo-devo, okay? And scientists have run into some extremely serious problems when they start looking into these things at a detailed level, okay? And we're going to look into that. So there's your sea urchin. So this is an article, Stephen Meyer's extensive treatment of evo-devo. This is from July 24th, 2024, on the Evolution News and Science Today website. We've been on this website before. So there's a lot here. We're gonna try and again wade through it. It's a lot. It's difficult, but again, I'm gonna try and break it down. And when it's fully understood, we're gonna see at the end of this that evolution is completely impossible. Evolution is completely impossible. There's no way it happened. It just could not have happened based on what we're observing with these regulatory networks. And that's not a creationist saying that. That's the people who are studying these things saying that, essentially. So we're gonna get through this, and we're gonna try and see. It's gonna be a lot. It's gonna be technical, but at the end of the day, we're gonna see that evolution is pretty much false. There's no way it could have happened. All right, so let's get started. Yesterday, I noted that Gerd Muller, emeritus professor at the University of Vienna, had responded last year to Stephen Meyer's points on the Joe Rogan podcast about development and evolution. I guess it's Muller, whatever it is. Muller is a critic of neo-Darwinian theory and its ability to explain the origin of biological novelty, and instead prefers evo-devo-based models to explain how new body plans arise. While this did not come up much in the Rogan podcast, Meyer in Darwin's Doubt actually wrote quite a bit about evo-devo-based models of organismal evolution. Settle in for a long read. Yes, we're going to have to go through all this. And I think we'll touch on these again because they're so important for understanding why evolution is false. Prepare for a long read because in the book, Meyer devotes a lot of space to the subject. Here's what he writes. Evolutionary theorists and developmental biologists such as Rudolf Raff, Sean Carroll, and Wallace Arthur have developed a subdiscipline of biology known as evolutionary developmental biology, or evo-devo for short. The evolutionary developmental biologists have since formulated alternative models that challenge a key aspect of neo-Darwinian triad, whereas neo-Darwinism envisions new form arising as a result of slow incremental accumulations of minor mutations. Evolutionary development biologists argue that mutations affecting genes involved in animal development can cause large-scale morphological change and even whole new body plans. One approach falls in the rubric of evo-devo and conceives of mutations producing modifications in larger increments. The neo-Darwinian synthesis has long emphasized that large-scale macroevolutionary change occurs as the inevitable byproduct of the accumulation of small-scale microevolutionary changes within populations. The consensus in support of this idea began to fray in evolutionary biology during the 1970s when young paleontologists such as Gould, Niles Eldridge, and Stephen Stanley realized that the fossil record did not show a pattern of gradual micro to macro change. Now, that's important. This is not what we're going to discuss, the fossil record, but there are some very famous scientists who looked at the fossil record and say, this just does not show evolution. At least not in the way Darwin proposed, which is that it would be slow changes over time. They see countless types of creatures just appearing full-blown in the evolutionary record with no discernible evolutionary antecedent. And these are people who believe in evolution, and what they're basically saying is somehow evolution happens very quickly. And it doesn't happen slowly like Charles Darwin said, because a lot of the fossil record is things just appearing out of nowhere. So, and again, that's filtering it through the idea that these things evolved at all, of course. From a creation standpoint, it makes complete sense. But again, it depends on your perspective. But these evolutionary biologists recognize the problem. In 1980, at a now famous symposium on macroevolution at the Field Museum in Chicago, the rebellion burst into full view, exposing what developmental biologist Scott Gilbert called an underground current in evolutionary theory among theorists who had concluded that macroevolution could not be derived from macroevolution. So, uh, uh, you don't know evolution can be disproven in step 1 and step 2 and 5, but maybe it can. Well, it's helpful to see all this, Lionheart. Because the more people see that there are these real scientific issues, the more that they will be convinced. I mean, there are arguments all over the place, but I'm just presenting one argument here. At the conference, paleontologists who doubted the micro to macro consensus found allies among younger developmental biologists. They were dissatisfied with neo-Darwinism, in part because they knew that population genetics is mathematical expression. No, this is we're not going to be. It's not a discussion today, Lionheart. You can just join some other time. I'm just going to try and run through this. Sought only to quantify changes in G frequency rather than explain the origin of genes or novel body plans. Thus, many developmental biologists thought that neo-Darwinism did not offer a compelling theory of macroevolution. To formulate a more robust theory, many developmental biologists, such as Rudolf Raff, a developmental biologist at University of Indiana and one of the founders of evo-devo, urged evolutionary theorists to incorporate insights from their discipline. For example, developmental biologists know. Just post it in the chat, Lionheart. Just explain it. For example, developmental biologists know that mutations expressed early in development of animals are necessary to alter body plan morphogenesis. Thus, they argue that these mutations must have played a significant role in generating whole new animal forms during the history of life. They assert that this understanding of developmental process is crucial to understanding animal evolution. Some evo-devo advocates, such as Sean Carroll and Jeffrey Schwartz, have pointed specifically to homeotic or Hox genes, master regulatory genes that affect the location, timing, and expression of other genes, as entities capable of producing large-scale change in animal form. These evo-devo advocates have broken with classical neo-Darwinism, primarily in their understanding of the size or increment of mutational change. You're boring your audience away, dude. Up to you. Yeah, I really don't care if the audience is bored. I'm trying to explain this topic, and you can't really do it without being boring. So the people who are interested, really in truth, will stick around. The others who don't really care will go away. I can't be concerned about that. But anyways, basically, so what we've read so far is that scientists are recognizing not only that there are problems with the conventional theory of evolution as it's presented in the mainstream, but also that these developmental pathways must be incorporated into whatever evolutionary theory is put forward. So basically, they're recognizing that these developmental pathways, these gene regulatory networks, must change, and potentially can change so rapidly that they create whole new body plans very quickly, which is what the fossil record puts forward. But we're gonna get it. So now that's just the background. Those aren't really the problems. The problems come up later. The problems come up when we start looking at these developmental pathways and seeing why this can't really be the case. Major but not viable, viable but not major. Despite the enthusiasm surrounding the field, evo-devo fails, and for an obvious reason. Its main proposal, that early acting development mutations can cause stably heritable large-scale changes in animal body plans, contradicts the results of 100 years of mutagenesis experiments. And here's where the problems arise. The experiments that they've done on these networks show that this can't happen. So what are we talking about? As we saw in chapter 13, again, this is in reference to Darwin's doubt, the experiments of scientists such as Nuslin, Volhard, and Weishaupt have shown that definitively that early acting body plan mutations invariably generate embryonic lethals. So here it is right here. This is what we're going to get into. There are scientists who have recognized this issue about developmental networks, about gene regulatory networks. They did experiments on them, and what did they find? Every single time that they tried to mutate these networks, it killed the organism. And one thing we're going to see is that they were so determined to do this that they fully basically they found every single gene in some of these organisms. Some of them were pretty simple organisms, and what they basically said was, we're going to mutate every single one of these genes until we get a different creature. What they found was, and we're going to continue to look at this, that there were some genes which were essential to the development of the organism, which obviously would need to change for it to go to a different organism, that when they mutated them always killed the organism. And this leads to a roadblock for evolution, because you have to change these networks, but if you change them, you kill the organism, because they're controlling essential parts and organs of the organism itself. Dead animals incapable of further evolution. The results of these experiments have generated the dilemma for evolutionary biologists that geneticist John MacDonald aptly described as the Great Darwinian Paradox. Recall that MacDonald noted that early acting regulation mutations do not produce viable alterations in form that will persist in populations, as evolution absolutely requires. Instead, these mutations are eliminated immediately by natural selection because of their invariably destructive consequences. On the other hand, later acting mutations can generate viable changes in the features of animals, but these changes do not affect global animal architectures. So basically what's being said here is that when you basically eliminate the early acting regulatory genes in these organisms, it creates a catastrophic defect for development and kills the organism. But if you mutate the ones that are activated later, that control less important aspects of the organism's development, then you can get sort of types of variation. And that makes perfect sense based on what you observe in nature. Think about dogs. We can imagine, just think about one aspect of dog, which is just the color of its fur. The genes that control the color of the fur are not as essential as the genes that control the development of the heart and the brain and the lungs and things like that. So the genes that control the fur, you can mutate them and manipulate them and do different things, and it'll produce a different looking dog or canine creature. But if you mutate the genes that control for the development of the heart or the brain and the lungs, you're going to kill the dog. Those things have to function. So and some of them, you can see some of them maybe like the length of the legs or the shape of the nose and the shape of the beak of a bird, the color of its wings, things like that. Minor things are things that can change, but the really important things that make the creature actually what it is can't change, or you kill the organism. So we're going to keep looking at this. So here we go. Again, we read this statement. This generates a dilemma. Major changes are not viable. Viable changes are not major. In either case, do the kinds of mutation that actually occur produce viable major changes of the kind necessary to build new body plans. So to build a new body plan, let's think about building a new body. You're trying to get from a fish, for instance, to an amphibian. You're going to have to make a lot of changes to the body plan. Some amphibians have gills, but a lot of amphibians have lungs. So let's think about that. You're going to have to change from having gills to having lungs. That's a pretty huge change in the developmental pathway of the organism. What we're finding essentially is that when you mutate for instance, the genes that control the development of the gills and things like that, you can't change them. You can't go anywhere. You can't get from a fish with gills to something that doesn't have gills, because when you try to mutate that, but the problem is, if you try to get from a fish to an amphibian with lungs, then you have to do that. So how does it work? This is the question that's being raised. This is a question that's being asked. We're going to skip down for time's sake. Again, you can go and read this on your own. Some of this has to do with responses by evolutionary biologists trying to explain away what's going on, which is important. And I'm sure they would like me to read that stuff, but basically at the end of the day, they don't have any answers. That's just my take on it. You can go verify that yourself. But there's a lot of speculation about how things might have been in the past, but very little to explain what we actually observed, which is that basically what we're observing shows that evolution is actually impossible. There's no way it happened. It can't have happened. Everything stuck is what it is, except within within limits. Okay, so this is what the science shows. There's just no way around it. Stephen Meyer on dGRN's. At this point, it's worth recounting what Meyer writes about dGRN's in Chapter 13 of Darwin's Doubt. Developmental gene Now, this again, we're wading into the weeds here. Again, like Aaron Lionheart said, this can be boring stuff until you realize its implications that show that it basically proves nothing can evolve. Nothing can go from one kind of an organism to another. It really can't happen, which is very exciting because a lot of people believe that it can happen, and they're dead wrong based on actual science, not creationist talking points. And these aren't creationist scientists saying this. These are actually people who basically uphold evolution despite what their research shows, despite what their own research is saying. They are the ones saying that these experiments show that these pathways can't evolve, even though that would be necessary for evolution to happen. So we already read all this. So let's get into an example of someone who did study this his entire life. His name is Eric Davidson. He's a scientist. And full disclosure, he was someone who believed in evolution, and I say was because he passed away a few years ago. But he was probably one of the preeminent evolutionary biologists studying this field of evo-devo, and he came away with some pretty startling conclusions, despite the fact that he believed in evolution. As far as I know, up to the day he passed away, he actually basically admitted that the current science does not support that. And we'll see that in a little bit. No biologist had explored the regulatory logic of animal development more deeply than Eric Davidson at the California Institute of Technology. Early in his career, collaborating with molecular biologist Roy Britton, David formulated a theory of gene regulation for higher cells. By higher cells, Davidson and Britton meant the differentiated or specialized cells found in any animal after the earliest stage of embryological development. Davidson observed that the cells of an individual animal, no matter how varied in form or function, generally contain identical genomes. So basically, the brain, the lungs, the heart, the stomach, the skin cells, they all have the same genes, but they all turn into different cells. So how does that work? Well, this is their theory. During the life cycle of an organism, the genomes of these specialized cells express only a small fraction of their DNA at any given time and produce different dRNAs as a result. These facts strongly suggest that some animal-wide system of genetic control functions to turn specific genes on and off as needed throughout the life of the organism, and that such a system functions during the development of an animal from egg to adult as different cell types are being constructed. So this is obviously very straightforward. You hardly have to be a scientist to predict this, but basically all our cells have the same DNA, the exact same DNA. So what is it that makes a brain cell different from a lung cell, different from a heart cell, different from a kidney cell, different from everything else in our body? And what's going on is that in our cells, different parts of the genome are being activated at different times, which lead to the differentiation of the cells. When they proposed their theory in 1969, Britton and Davidson acknowledged that little is known of the molecular mechanisms by which gene expression is controlled in differentiated cells. Nevertheless, they deduced that such a system must be at work. Given that tens or hundreds of specialized cells arise during the development of animals, and that each cell contains the same genome, they reasoned that some control system must determine which genes are expressed in different cells at different times to ensure the differentiation of different cell types from each other. Some system-wide regulatory logic must oversee and coordinate the expression of the genome. I'm reading quickly just because this is just background information before we get to the conclusions. Davidson has dedicated his career to discovering and describing the mechanisms by which these systems of gene regulation and control work during embryological development. So he specifically focused on embryological development. During the last 2 decades, research in genomics has revealed that non-protein coding regions of the genome control and regulate the timing and expression of the protein coding regions of the genome. So this is exactly what they predicted. They found that the parts of the genome that don't code for DNA, scientists used to believe that only the DNA coding the protein coding section of DNA was functional, and everything else was quote unquote junk DNA. Now that was basically premised, believe it or not, on evolutionary predictions that there's all this junk DNA from when we were monkeys and fish and pond scum that's just left in there and just got, it's all just a bunch of junk now. It doesn't do anything. But the show is where the proteins are being coded. That's what makes us a human or a bird or a tree or whatever we are. Turns out evolutionary scientists were completely wrong, 100% wrong. The parts and what stands to reason, as we might predict, the parts of the genome that don't code for proteins are regulating the parts of the genome that do code for proteins. It's all functioning, just at different times and different places. It's all doing something. There are regulatory sections of the DNA, and there are protein coding sections. So evolutionists failed big time on that one. They didn't want to admit it. That was one of the major failed predictions of evolution. But there it is. And scientists, they this isn't something they admit as a failure. They're just like, oh yeah, we just discovered this. But if you go back, you will see evolutionary scientists saying that there is junk DNA and doesn't have any function. Now we know they were dead wrong. On arriving at Caltech in 1971, Davidson chose the purple sea urchin as the experimental model system. The biology of sea urchins makes it an attractive laboratory. I don't care about that. We're going to skip all this. Basically, they were looking for. They basically studied the sea urchin. That's all that was saying, and they were trying to figure out how it unfolds as an embryo. This process does not happen fortuitously in the sea urchin, but via highly regulated and precise control systems, as it does in all animals. Indeed, even in one of the simplest animals, the worm C. elegans, possessing just over 1,000 cells in its adult, is constructed during development by dGRN's of remarkable precision and complexity. In all animals, the various dGRNs direct what Davidson describes as the embryo's progressive increase in complexity. An increase, he writes, that can be measured in informational terms. Davidson, so this is where we start getting into the more interesting stuff. Davidson notes that once established, the complexity of the dGRNs as integrated circuits makes them stubbornly resistant to mutational change. Ooh, look at that. See, scientists, there's a lot of people out there who want you to think that there's no reason to think there's just this limitless, unbounded potential for evolution to just work on creatures, and there's nothing holding it back. That's not what science has discovered. There are parts of the DNA which are, quote, stubbornly resistant to change, and that's quite an understatement, as we're going to see. A point he has stressed in nearly every publication on the topic over the past 15 years. In the sea urchin embryo, he points out, disarming any one of these substructures produces some sort of abnormality in expression. Developmental gene regulatory networks resist mutational change because they are organized hierarchically. This means that some developmental gene regulatory networks control other gene regulatory networks, while some influence only individual genes and proteins under their control. At the center of this regulatory hierarchy are the regulatory networks that specify the axis and global form of the animal body plan during development. These dGRNs cannot vary without causing catastrophic effects to the organism. There you have it. These dGRNs cannot vary without causing catastrophic effects to the organism. What's worth noting at this point is that all organisms have different dGRNs, which is why they lead to different organisms. So, if dGRNs cannot vary without causing catastrophic effects to the organism, how do you get from one organism with one set of dGRNs to another organism with another set of dGRNs? This is the grand question. This is the grand paradox that we already discussed. There is no way to get from one organism to another. There's none, based on these scientific experiments. Indeed, there are no examples of these deeply entrenched, functionally circulates varying at all. You cannot vary them. You cannot introduce mutations to them to make them go to something else where they will create a functional organism, as far as we have observed. And at this point, there's no reason to think we will observe anything else, because at this point, scientists have studied a number of creatures. They've sequenced all the genomes. They've found all these regulatory networks. They've mutated them. And they have not found a single case in which these regulatory networks can be changed or modified in any way. It always leads to the destruction of the organism. This, my friends, absolutely destroys evolutionary theory. Evolution is a dead theory. There's nothing left to it. Just this fact alone destroys evolutionary theory. So that needs to be understood. Now, the public isn't aware of this because they're not being taught it essentially. But that's why his philosophy is here, and that's why I'm actually just reading other people's work, so I can't really claim credit for figuring these things out. But I'm just trying to promote this so that more people are aware of what's going on. That evolution's dead. It's impossible based on scientific experiments, not creationists. It's not someone reading the Bible and saying God did it in 6 days. No, these are evolutionary biologists who are creating this data and showing these things. At the periphery of the hierarchy are gene regulatory networks that specify the arrangement of smaller scale features that sometimes vary. Okay, so there are some of these genes that when you mutate them can mutate and which produce variations with it. And this is so big. This is so observationally based. This is what we actually observe in nature. There are variations within kinds, but we do not observe kinds changing into other kinds. We basically, think about dogs. The dogs seem like to be one of the most elastically variable creatures we see out there. We have Great Danes and we have Chihuahuas. But the body plan is essentially the same. You have the head with the snout and the teeth and the vertebrae and it's all kind of you can't get there, but you're not going to get from a dog to something other than a dog. You're going to get like a variation of a dog body plan. Because if you try to mutate some major aspect of the dog, like let's give an example. Mammals supposedly evolved from reptiles. Okay. So, or maybe there's another one. Let's think of another one. Yeah, I think birds evolved from reptiles. Yes, I think that is what they claim. So get to a bird. There's all kinds of really massive changes to the body plan that have to take place. There's differences in the organs. Or wait, is it? Yeah, mammals and birds both evolved from reptiles, according to this evolutionist. There's all these giant changes that have to take place. There's fundamental changes to the structure and the layout of the body plans and things like that, and the organs. There's different organs and things like that. You have to be able to change the gene regulatory networks that control these things, but you can't really do that. You have to get from the reptile dGRN to the bird dGRN or a mammal dGRN, but the dGRN of the reptile is stuck as what it is. It can't change. You can't change it. So it's stuck as a reptile. It may be like a smaller reptile, a bigger reptile, a red reptile, a green reptile, whatever. But it's going to be a reptile no matter what, because the dGRN is controlling it to go into that direction in its embryo. So if you can't change the dGRN, you can't change it from reptile to a mammal. You can't change it from a reptile to a bird. You just can't. But you can change it from a reptile with short claws to a reptile with long claws and things like that. So all this is perfectly consistent with what is observed in nature. This requires mutating the varying circuits that do not vary without catastrophic effects. As Davidson emphasizes, mutations affecting the dGRN that regulate body plan development lead to catastrophic loss of the body part or loss of viability altogether. He explained. Okay, so this is Eric Davidson. He's an evolutionary developmental biologist. He is the one saying that when you mutate these dGRNs, that is what causes loss of viability. This is not a creationist saying this isn't creationist. These are real scientific observations that no one can deny. Atheists, creationists, whoever. All right. He's the one who did this research, and among others, saying this. When they try to mutate these dGRNs, it kills the organism. You can't get it to change from what it is. There is always an observable consequence if the dGRN subcircuit is interrupted. Since these consequences are always catastrophically bad, flexibility is minimal. And since the subcircuits are all interconnected, the whole network partakes of the quality. But there's only one way for things to work. And indeed, the embryos of each species develop in only one way. Wow. That's a quote from Eric Davidson, the man who's spent decades of his life studying these things. I think it's worth reading again. There is always an observable consequence if a dGRN subcircuit is interrupted. since these consequences are always catastrophically bad, always catastrophically bad, flexibility is minimal. And since the subcircuits are all interconnected, the whole network partakes of the quality that there is only one way for things to work. And indeed, the embryos of each species develop in only one way. So what do we learn here? The dGRN controls the development of the organism. If you mutate the dGRN, which would be necessary to change for it to evolve into something else, then you kill the thing that's developing. So you can't mutate it. And that means that there's only one way for things to happen. And this is true for all species that have been observed. Therefore, everything is stuck as what it is. Evolution is very much has restrictions. There are some things that cannot evolve. You can evolve peripheral things, secondary things like hair color and height and minimalistic things like that. But when it comes to the fundamental body plan of the organism, you can't change it. But that would absolutely be necessary to get from a fish to a reptile to an amphibian to a bird to a human or a mammal or whatever it is. Everything's stuck as what it is. This absolutely does falsify evolution. There's just no other way to look at it. Davidson's findings present a profound challenge to the accuracy of neo-Darwinian mechanism. Building a new animal body plan requires not just new genes and proteins, but new dGRNs. This is what a lot of people don't understand. A lot of people think about evolution as just new genes and new proteins. They don't realize that a lot of what's going on in the genome is regulation of everything that's going on in the body. If you can have basically the same protein coding regions. Let's say humans and chimps have. This isn't true, but let's just say humans and chimps have the same protein coding regions, which they pretty much do. For them, I mean, like 99% similar in a lot of cases. The reason a human isn't a chimp and a chimp isn't a human is because of the dGRNs. The dGRNs of a chimp make it a chimp, and the dGRNs of a human make it a human. That's where the differences lie. And we're actually going to look at this early. I just want to kind of briefly show this. If we have time, we'll get to this. Bombshell. New research overturns claims that humans and chimps differ by only 1% of DNA. So if we have time, we'll get around to that. I'm not sure we will, but that is something to look forward to if we have time. But what I'm trying to highlight here is essentially that everything is stuck is what it is. You need to if evolution requires the evolution of one gene regulatory network to another. That's what actually creates the shape of the organism as it develops as an embryo. So if you don't develop new gene regulatory networks, you won't develop a new organism. That's a scientific fact. But the problem is every time scientists try to mutate these things, it doesn't work. It just kills the organism. So this is a serious problem for evolution. And you don't hear a lot of evolutionists talking about it on the internet. I look at their channels. I hear them talking about the evidence for evolution. And sure, there's some evidence, but there are defeaters for evolution too that they don't present their audiences, and a lot of people don't know about them. But here we have one right here where, and by the way, I want to let people know, scientists, I'm aware of this. Scientists have been trying to create new creatures in labs for a long time. They have been trying to take one organism and change it into another organism since basically Darwin proposed this theory, because some of them desperately want to prove his theory correct. And they want to do it by taking these organisms and changing it. They have not succeeded yet. There is not one case of scientists taking a type of organism into a lab and changing it into something completely different that's viable and which is a different population, okay? And the reason for that is because it's impossible. Because they've tried desperately for so long, and what they keep running into are these walls. Evolution has walls. It has limits. So they will get variations. They will get strange mutations. They will get all these different kooky little things that they create, but they're not getting anything that is basically like, changing from a worm to, a beetle or something like that, which is what they would love to show. But to build a new dGRN from a pre-existing dGRN by mutation and selection necessarily requires altering the pre-existing developmental gene regulatory network. The very kind of change that we saw in Chapter 12 cannot arise without multiple coordinated mutations. In any case, Davidson's work has also shown that such alterations inevitably have catastrophic consequences. Okay, we're almost done. Let's try and get through this. Davidson's work highlights a profound contradiction between the neo-Darwinian account of how new animal body plans are built and one of the most basic principles of engineering, the principle of constraint. Engineers have long understood that the more functionally integrated system is, the more difficult it is to change any part of it without damaging and destroying the system as a whole. Davidson's work confirms that this principle applies to developing organisms in spades. The system of gene regulation that controls animal body plan development is exquisitely integrated so that significant alterations in these gene regulatory networks inevitably damage or destroy the developing animal. But given this, how could a new animal body plan and the new dGRNs necessary to produce it ever evolve gradually via mutation and selection from a pre-existing body plan instead of dGRNs? Davidson makes clear that no one really knows. Contrary to classical evolutionary theory, the processes that drive small changes observed as species diverge cannot be taken as models for the evolution of body plans of animals. He elaborates. This is Davidson, the evolutionary biologist, elaborating. Neo-Darwinian evolution assumes that all process works the same way so that evolution of enzymes or flower colors can be used as current proxies for study of evolution of the body plan. It erroneously assumes that change in protein coding sequence is the basic cause of change in developmental program. And it erroneously assumes that evolutionary change in body plan morphology occurs by a continuous process. All of these things fit within evolutionary predictions, but what we actually observe is that the exact opposite is true. Evolution is being torn asunder by these observations. All of these assumptions are basically counterfactual. Wow. The basic fundamental necessities of evolution are counterfactual. That's an evolutionary scientist saying that, not a creationist. This cannot be surprising since the neo-Darwinian synthesis from which these ideas stem was a pre-molecular biology concoction focused on population genetics and natural history, neither of which has any direct mechanistic import for the genomic regulatory systems that drive embryonic development of the bioplan. So what he's basically saying is that when Darwin and his fellow kinsmen developed the theory of evolution, it was based on the science of that time. But obviously, the science developed, more discoveries were made. Now they have to incorporate all this information that wasn't known at the time about molecular biology. And unfortunately for evolutionary scientists, the molecular biology is demonstrating that evolutionary change is completely impossible. There is no way, based on current observations, which are quite extensive and I would say perhaps exhaustive at this point. Well, they haven't mutated every single organism on Earth and sequenced all the genomes, so they haven't done. But for the organisms that have been studied, which include simple ones and more complex ones, every single time that scientists look at these regulatory networks, there is no way to mutate them, which means that indeed evolution has hard limits. You can get variation within a species, within a kind, but eventually you will hit a wall and you cannot get to a different organism. But that would have obviously been necessary for evolution to occur. You need to get from basically a simple worm-like organism or single-celled bacteria to whatever was after that, worms or whatever it was, from worms to crustaceans, again, or sea slugs or whatever they were, to fish, to amphibians, to reptiles. All of these things require a change in these gene regulatory networks. But unfortunately, we are finding that according to modern science, that's completely impossible. Let's go a little bit forward. We have a little more time. In a post yesterday, we saw that this is a second article on developmental gene regulatory networks, the scientific literature supports Stephen Meyer. And this is from July 25th, 2024 on Evolution News website. If you want to go look at that yourself. I'm not going to take too much time just because I feel like perhaps people are getting bored. I don't know how anyone could get bored by this. I think it's incredibly fascinating just seeing that evolution is absolutely false. There's really no doubt about it based on the science we have before us. But that's the interesting fact is they will tell you that the science we have before us all supports evolution. Actually, no, the science we have before us refutes evolution. It shows it's impossible. That doesn't mean there won't be new science in the future that maybe helps evolution, but what we have right now shows evolution's impossible. We saw that Stephen Meyer wrote extensively about evo-devo and Darwin's doubt, effectively answering biologist Gerhard Mueller's preferred evolutionary model for how new body plans arise. If I could boil down Meyer's arguments to 3 points, they would be: evo-devo focuses on the role of special early acting mutations in developmental processes to generate new body plans, but over 100 years of mutagenesis experiments show that mutations in genes regulating development are invariably deleterious, in some cases having only trivial effects. Meyer summarizes this generates a dilemma. Major changes are not viable, viable changes are not major. In neither case do the kinds of mutation that actually produce viable major changes of the kind necessary to build new body plans. We see these deleterious effects particularly in experiments on developmental gene regulatory networks, complex networks of gene interaction which regulate the expression of genes early in development as an organism's body plan begins to grow. After reviewing experimental work on dGRN's, Meyer finds that these dGRN's cannot vary without causing catastrophic effects to the organism. We saw that earlier with the work of Eric Davidson. These experimental results on dGRN's have profound implications for organismal evolution, because if changes of dGRN's are lethal to an embryo, how can they be modified to explain how new body plans evolve? This is the big question right here. And the answer is they can't. Meyer writes in the book, the system of gene regulation that controls animal body plan development is exquisitely integrated so that significant alterations in these gene regulatory networks inevitably damage or destroy the developing animal. But given this, how could a new animal body plan and the new dGRNs necessary to produce it ever evolve gradually via mutation and selection from a pre-existing body plan instead of dGRN? These are really difficult questions for the evolutionist based on the scientific data. As it stands right now, the scientific data is showing that evolution is completely impossible. Horribly impossible. Beyond impossible. Like, there's nothing to support the idea that organisms can evolve from one kind to another, which leads to the question, how did that happen? If you can't get from one kind of organism from another, then how did all these organisms arise? How did they get all these extremely coordinated circuits of gene regulatory networks out of just random blind mutations that can't change? Did atoms, if there is a skeptic out there, a materialist and atheist, you have to assume obviously that there was no mind involved in these, because atoms and molecules don't have minds. They had to just move around in space and figure all this out just from bumping around in each other. But atoms and molecules don't care where they combine or what shape they're in. They don't care if they're in the shape of a bird, or half a bird, or the wing of a bird, or the DNA of a bird, or if they're in the shape of a rock, or if they're in a drop of water, or if they're in a star or moon dust or whatever it is. They don't really care to form themselves into these complex circuits of gene networks that are necessary for these organisms to survive. That stands today, and that stands during the course of... So how do you propose that these mindless molecules moving around in space were able to accomplish all this in the history of Earth? And you realize there's a time limit here. In some cases, we have animals evolving in the course of only 10 to a few dozen million years. Oh, 10 million years? That's a long time. Not really. Not when you consider all that these mindless molecules have to figure out to get from one organism to another. When our smartest scientists can't even mutate a single-celled organism into something other than what it is. Okay, these are real problems for evolution. Uh, uh, Basically, there are some scientists who are. There are many scientists who are aware of this. There's some scientists who are responding to this. Okay, let's look a little bit more at Eric Davidson works, a scientist who really pioneered a lot of this. There is a high penalty to change in dGRN's in that interference with the dynamic expression of any one of these genes causes a of collapse of expression of all. Look at that. There is a high penalty to change in dGRN's in that interference with the dynamic expression of any one of these genes causes the collapse of expression of all. Imagine trying to evolve a new creature, and if you mutate one of these genes, it destroys all the others. And the total loss from the system of their contributions, and by the way, you have to do that. All organisms have different dGRNs. So you have to mutate the dGRN. But if you mutate the dGRN, you kill the organism. How are you getting from one organism to another? And the total loss from the system of their contributions to the regulatory state. There is always an observable consequence of a dGRN subcircuit interrupted. Since these consequences are always catastrophically bad, we already saw this, but let's read it again. Flexibility is minimal, and since the subcircuits are all interconnected, the whole network partakes of the quality that there is only one way for things to work. And indeed, the embryos of each species develop in only one way. Wow. There it is right there. That's evolution being killed in a really horrible way and not coming back. A few years ago, remarkably conserved subcircuits termed network kernels that operate high in the dGRN hierarchy were discovered. These kernels similarly canalize downstream developmental process in each member of each given clade. All right. Evolutionary inflexibility due to highly conserved canalized dGRN kernels. As discussed above, these subcircuits operate at the upper levels of dGRN hierarchy, so as to affect characters of the body plan that are definitive for upper level taxa, i.e. they control the early stages of just the types of developmental process of which the invariance per taxon constitutes our problem. So what does all this mean? Basically that there are dGRNs which there are some that are more important than others, and these important ones control the early stages of these various taxa. Since they preclude developmental alternatives, they may act to Booleanize the evolutionary selective process. Either body part specification works the way it's supposed to, or the animal fails to generate the body part and does not exist. Since they preclude developmental alternatives, meaning they can't develop any other way than what they are, they may act to Booleanize the evolutionary selective process. What does this mean? What that means, essentially, is that they act to solidify the evolution of they make it impossible for the organism to be other than what it is. Booleanize, this is just fancy language. This guy is trying to hide what's going on because he knows that the entire apple cart is being this is really complex language saying things can't vary, and what these things are doing is they're making it so that everything is basically fixed as what it is. He's admitting it, he's being honest, but he's doing it in a way that's kind of confusing to really interpret. The body part the specification must work the way it's supposed to, or the animal fails to generate the body part and does not exist. Eric Davidson, Evolutionary Biosciences, Regulatory Systems Biology, Developmental Biology, 2011. There it is right there. There's your citation. He's the one saying that these dGRNs are fixing all organisms as what they are, and they are stopping evolution from occurring. Or this, interference with expression of any genes in the dGRN kernel by mutation or experimental manipulation has severe effects on the phase of development that they initiate. This accentuates the selective conservation of the whole subcircuit on pain of developmental catastrophe. Again, this is just very complicated language saying when you try to mutate these things, it kills the organism. You can't mutate them. These things can't mutate. So all these proponents of evolution on YouTube and in the scientific community are absolutely lying when they say there are no limits to evolution. Because scientists have known for decades that there are very strict limits to evolution based on exactly what we're learning about right here, which I'm telling you. Evolution cannot occur in an unlimited fashion. This is very well. This is observed science. This is exactly what all of our scientific experiments are showing. That when you try to mutate these genes, and that's exactly why, as I said earlier, whenever scientists try to go into a lab with one creature and come out with another, they can't do it. You would think by now they would have done that. All these evolutionary biologists working for decades to prove evolution, they can't create one new creature from a pre-existing one. They can't even take E. coli and turn it. It's a little rod-shaped single-cell bacteria. Do you realize all E. coli look exactly like all the other E. coli? And they've been trying to mutate this little single-cell bacteria for decades, and they can't get it to change even a little bit. And there are literally E. coli multiply way faster than humans because they're bacteria. And there are way more of them. There are trillions and trillions of bacteria on a little petri dish. And they all look exactly the same, and they don't mutate into anything else. And they want to say that whales evolved from like a little dog-like creature in less than 10 million years? When E. coli, we've been trying to mutate them and change them for decades? We can't get anything different? We can't even get them to look a little bit different from what they always were? See, this is really bad for evolution. Really bad for evolution. Good to see you, Von Eric. The intolerance of body plan affecting dGRN's to fundamental perturbations indicates that they could not have evolved by undirected mutations. Okay, this is where no longer a quote. This is the intelligent design proponent speaking now. Many coordinated mutations would be needed to convert one functional dGRN that generates a particular body plan into a different dGRN that generates. So basically, what's being said here, you would need to change the entire dGRN all at once. It can't happen piecemeal like evolutionary theory would dictate. Because if you mutate just one thing, then the entire system collapses. Here's the problem. You have to take an entire dGRN, which is built into the DNA organism, and completely replace it with a new dGRN with a new set of DNA. How could that happen in the history of evolution, which would require small mutations and changes over time? It can't happen. It's almost like an intelligent being created these dGRNs, exactly what they are, to produce the creature that is being created. It's almost like evolution can't do this, and it required some sort of intervention from an intelligent being that knew how to program all this stuff into the DNA from the beginning. That would actually make way more sense than thinking this happened within the time frame of Earth's history, even if you give Earth billions of years of existence, as scientists do. And maybe it is billions of years old. Even if it is, it doesn't really seem to solve these problems because it's not about time limits. It's about functional constraints within the genome. You can't, even if you gave it trillions of years, these things apparently can't change. So 1,000 years, a billion years, it's all the same. It's not going to change no matter how long you give it. Okay, so here I'm not going to read this. Basically, there are scientists who have answered these problems. Let me think a little bit about this. This is Eric Davidson. This is Eric Davidson again. Deconstructing the evolutionary process by which stem group body plans are stepwise formulated will require us to traverse a conceptual pathway to dGRN elegance, beginning where no dGRN provides a model. So he's just admitting that there's no dGRN they've observed that can change. The basic control features of the initial dGRNs of the pre-Cambrian and early Cambrian must have differed in fundamental respects from those now being unraveled in our laboratory. So what he's saying here, this is the cognitive dissonance of an evolutionary biologist. This is what he's saying. He's basically saying, for evolution to have occurred in the past, these dGRNs must have operated completely differently in prior organisms different from the ones we're studying now. The earliest organisms must have had very flexible dGRNs. Now, that assertion is fully and totally based on the presupposition that evolution must have occurred at all. why would we assume, based on all the organisms we've studied so far, that the dGRNs of these early creatures were any different from the ones now? That we have every reason to suspect that the dGRNs of early organisms function just like the ones that do now, and they can't change. But what he's basically saying is, yeah, it looks bad for evolution right now, but early on in life's history, everything was really elastic and flexible, and that's how everything got to where it was, and now we're just kind of messing around the edges. But there's no reason to suppose that. There's no reason to. That's just that's speculation. That's wishful thinking. It's guesswork. It's not scientifically rigorous. It's not something that's scientific. It's just basically an evolutionary biologist trying to create a narrative that suggests that evolution was possible in the past, even if it isn't now. And what he's basically saying is, yeah, evolution's stuck now. We can't evolve right now. But in the past, things were able to evolve much more rapidly, which, is not scientific. It's not based on observation. It's based on philosophical thinking. I would say religious thinking. Paleontologist Charles Meisel said much of the statement in 2013 when responding to journal Science and Meyer's arguments and Darwin's doubt regarding dGRNs. Marshall argued that although Meyer is correct to observe that manipulation of such networks is typically lethal, this is not a problem for evolution because GRNs at the time of the emergence of the phyla were not so encumbered. So this is their response. If you want to know how scientists are responding to this information, this is essentially what you're getting, largely or entirely. They're all saying that, okay, the experimental data we have now shows that GRNs are immutable, which means evolution is impossible as we currently exist. But millions of years ago, creatures were simpler, and even though we are unable to study their genetic regulatory networks, we can basically surmise that their networks were more flexible, which permitted for more evolution, which permitted for life to get to where it is, and now it's kind of stuck. But again, this isn't something they've actually observed. This is just something this is just something they're saying to make it look like evolution, at least at one point, was possible, even though there's nothing that they've observed that demonstrates that. So this is their religious belief taking over. The idea that this is their faith. This is their faith. Sure, we don't observe what it is we believe, but it must have been the case because we know evolution happened. It must have happened because how else could life get here unless it evolved? Right, exactly. That's the question. They are holding on to the idea that life evolved, and for life to evolve, things must necessarily have been different than they currently are. That is not science. These people are abandoning science in favor of their faith and their religion. Again, I think we can end it there. There's a lot more here. It only gets worse for evolution. There's a lot more to talk about, but I've kind of already gone over the time limit I wanted to. So I showed you the articles. I'll post them in the link or the description. I'll post the links in the description if you want to go review them for yourself, just to make sure I'm not taking anything out of context. But I think I've reviewed enough to show that there's serious problems here for evolution, regardless of how the evolutionary biologists are responding. They're basically providing nothing except speculation and wishful thinking, which isn't based on science. I did have kind of a fun article here that I was going to put up, but I'm not sure if time will permit. This is from actually just like a week or two ago. There's a research paper that came out, again, not from creationists, that basically destroyed the lie that's been promoted for so long that humans and chimps share 99% DNA. It's actually not even close to that. What people don't understand is that the 99% of DNA is only the protein coding regions. When you look at the rest of the chimp DNA and human DNA, compare it, there's massive, massive differences. It's actually closer to like 85% similar, which just sounds shocking because, we've been lied to so long and told it's 99%. But that lie was based on only looking at a small portion of the genome. New data in a recent published Nature paper by you. So these are not creationists who are promoting this information. Again, a lot of people know that humans and chimps are not 1% DNA. This is a cultural myth that is promoted by scientists who are lying to the public. It's not true. Humans and chimps do not share 99%. It's way different. Way different. There's some. So basically, in this paper, and I'm not going to go into it just because I don't want to take too much time, but what they found when they compared humans and chimp DNA, at least 12 to 15%, at least 12.5%, and possibly up to 13.3% of the chimp and human genomes represent a gap difference between the two genomes. That means there's a gap in one genome compared to another, where they're so different they cannot even be aligned. There are also significant alignable sections of the two genomes that show short nucleotide variations, which differ only about 1.5%. Okay, so that's the 1.5% right there. These are the parts of the genome that actually align, and which probably many of which code for proteins in humans and chimps. This is the lie. The lie is that this is the only part of the genome that matters, and this is what makes humans different from chimps. It's only 1.5%. But when you take the complete human and chimp genome, all the nucleotides together, there is a gigantic difference. Well, I mean, gigantic is relative, but it's much more than 1.5%. We can add the difference. So if you add up the non-alignable sections of chimp genome and the alignable sections, so you get 1.5 plus about 13, you're getting to about a 14 to 15% difference between human and chimp genomes, which is a gigantic chasm of a difference, which actually, again, is a huge problem for evolution based on the time frames we're given for human evolution. The reason is this: there isn't enough time in history for this kind of deviation to have occurred naturally. What do you mean? How do you know that? Well, because humans and chimps supposedly diverged basically at the same time. So they've been accumulating differences in their genomes for about the same amount of time. That just stands logic. That's just basic logic, okay? So if you look at the variation within the two groups, humans and chimps, there's not a whole lot of variation. So we can calculate the rate at which mutations are occurring within groups. The question becomes this: humans and chimps started at the same point, okay? So the differences between humans and chimps must have accumulated at the same rate as differences between intraspecies, essentially. So if you look at the rate at which, basically, take the human lineage, for example. Let's just say we branched off from chimps, I don't know what it is, like 5 million years ago, whatever they say it is. Since that time, we can look at all the humans on Earth and look at the differences between their genomes and say, this is the rate at which mutations are being generated in the human lineage. Same thing for apes or chimps, or which are a kind of ape. We can look at the time frame, the time that has elapsed, and we can say and look at the current genome between chimps and say there is this much variation within chimps. Therefore, we can say that the mutation rate was on average about this. So we know the rate at which things are mutating based on this timeline. When you actually look at the differences between, you have to ask the question, well, why is there such a massive difference between humans and apes compared to within the species? The number of mutations that have accumulated between the species is way more than has accumulated within the species, which suggests that for some reason the mutation rate is massively accelerated between the species. But how can that be the case if everything's operating at a uniform rate? Could it be that something accelerated the process? Could it be that there is no process, that these two types of organisms were just created as they are, as different organisms? And the reason there's not enough time to explain the differences between organisms is because time wasn't a factor. It was just a matter of them being created differently by some sort of intelligent being. The point is, the amount of time is not sufficient for this amount of of genomic difference to have occurred. And in any case, even if you disagree with that, the idea that humans and chimps are 99% similar is just simply a lie. It's just false. So regardless of how it bears on evolutionary theory, people who go around saying that humans and chimps are 99% similar are just plain wrong. They're just dead wrong. Again, this isn't creationists saying this. This isn't this isn't Answers in Genesis. This is actually a paper in Nature produced by scientists who purportedly, I would imagine, probably believe in evolution. They just are producing that a myth that is commonly believed wrong. Yeah, so here's the paper, Complete Sequencing of Ape Genomes, published April 9th, 2025, by Yu et al. Let's see if I can get to... I probably should have read this beforehand. Oh, this is interesting. Let's read this real fast. Look at this. This is pretty fascinating. Let's see if I can zoom in before my computer blows up. Stops working completely. Oops. Where was it? Oopsies. Let's go back. I lost it. Uh-oh. I'm gonna find it again and give it a try. Wait, how did I lose it like that? We'll find this real fast. We're gonna find it. We're gonna get this. This was interesting. Where on earth was it? Oh, here we go. Overall sequence comparisons among the complete ape genomes revealed greater divergence than previously estimated. Ooh, that's exciting. So if you look at these apes, you would predict a certain amount of divergence, and scientists for a long time thought that, there was this really similarities between all these genomes between these apes because they all look so similar. What happened? Indeed, 12 to 25% of an ape genome failed to align or was inconsistent with a simple one-to-one alignment, thereby introducing gaps. Ooh. Man. This isn't going to be reported. Not a lot of people are going to hear about this, but this is really bad for evolution. These scientists went in there and sequenced all these ape genomes, and they were expecting to find them all similar. Look at this. 12 to 27% ape genome failed to align. Oh my gosh. How is that possible? How do all these mutations arise so quickly within the ape lineage? Maybe it's because they didn't arise. Maybe it's because these apes were created the way they are. I don't know. I'm just throwing things out there. Anyways, you have this really huge problem for evolution. You have these really similar creatures. These scientists went in there. I'm sure they thought they were going to find all these similarities. Everything was going to match up. Nope, they didn't find that. Once again, evolutionary predictions were falsified. Gap divergence showed a 5-fold to 15-fold difference in number of effectable megabases when compared to single nucleated variant, which was due to, look at this. Look at this, which was due to rapidly evolving and structurally variant regions of the genome. And they know this how exactly? See, these are evolutionary biologists going in there, blowing up their own theory with facts, and then they're coming around and saying, actually, it's not a problem. You know why? Because there was rapid evolution. Yeah. That's what was happening. Instead of normal evolution, it was rapid evolution. Yeah. We'll use words to cover up the problem and deceive people into thinking evolution makes sense. That's what's going on there. And that's the same story. That's that. We might come back to that because I think that's pretty interesting. The fact that I might review that just because so many people believe in the lie that humans and apes share all this. There's no major. There are lots of people out there that have been fed the lie that humans and apes, or chimps at least, share 99% of their genome. It's not true. It's just simply not true. Those scientists proved it again. I've known this for years. A lot of people in the creation community have known this for years. But a lot of people who believe in evolution somehow still believe this lie, despite the massive amount of evidence that just proves it flatly wrong. All right, so we're going to end it here. We saw a lot of evidence tonight that evolution is completely impossible. A lot of evidence for failed predictions. We saw a myth get blown up in real time in the last couple of months by another scientific paper showing that humans and chimps don't share 99% of their genome. It's more like 85%. That's been known for a long. I first heard that probably over a decade ago. It was at the first when you first hear it, it's kind of surprising just because you hear the lie so much. But when you actually look at the data, yeah, humans and chimps don't share 99% of their DNA. It's vastly different. Humans and chimps don't even have the same number of chromosomes. So the people who say that it's all the same is they're really just misleading the public. Some of them are probably lying. I don't know what's going on, but some of them are scientists, which is the sad part. It's not just people on atheist YouTube channels promoting the lies. There are real scientists who are in on the action. I don't know why they feel the need to mislead the public like they do. Maybe they just don't know they're wrong. That's another possibility. But yeah, dGRN's. Again, to summarize, they are the basically the gene networks, the parts of the genome that code or program the development of the animal in its embryological stages. They are absolutely essential to the functionality. Or how are you going? You need these networks. You can't get from a zygote to a fully formed organism without them. And what science has shown is that they can't be mutated. They can't evolve. Which, if it's true, which it appears to be true, there's no reason to think it's false, means evolution has hard limits. You cannot get from one creature to another based on what we observe. Which begs the question, if we can't get from one type of a creature, now, again, more science might come along in the future that will help the evolutionists. But as of right now, it appears that they are being undermined by this data. So let's just assume the data stands. Let's just assume the current data we have is correct. Excuse me. How, if organisms can't evolve from one kind to another, which is what the data shows, how do we get all these different kinds of organisms? It's a puzzle. It's a puzzle if you believe in evolution. If you presuppose that evolution is how things came to be what they are. Now, a creationist can say, well, God created the DNA and he programmed the networks to be what they are, to produce the creatures they are. Now, there's no doubt that these networks are what produce the creatures that we observe. Like the network of a butterfly, you can imagine the dGRN being something different and it turning into like a beetle or something, or maybe even like, I don't know, something completely different. Maybe even like a rodent or something. I don't know. Who knows? The dGRN, the scientists for so long focused on the protein coding regions of DNA, they forgot, and that was because of evolution, said that the rest of the genome was just junk. Okay, they were wrong. The evolutionary predictions held us back for decades. They eventually found out that the rest of the DNA is doing things. Turns out it's involved in the regulation of the protein coding regions and the development of the organism. Okay. The protein coding regions are only a small player at this point. The reason an organism turns into the shape of what it is because of the regulatory networks. So the regulatory network is what produces a butterfly, differentiating it from a moth, differentiating it from a beetle, differentiating it from a worm, differentiating it from whatever. All these things are stuck as what they are, based on the current scientific data. You can't, and again, how do we know that? Or what is a good reason to believe that? Let me put it that way. The reason is because scientists have been trying to change these things for so long, we've never gotten anything different. We have sequenced the entire genomes of single-celled bacteria. Every single nucleotide. We know what it is. We know what it does. And they have tried desperately to turn these single-celled worms into something other than a single-celled worm. Even a different-looking single-celled worm. And they have completely failed. And the reason is because of these dGRNs. They can't mutate them. There are parts of the genome of every single creature which are immutable. Now, there are parts that are mutable and they produce mutant variations, but basically the point is you hit a hard wall, a hard limit. So this is this falsifies evolution as far as we know. And I say that because who knows if more data will come along and somehow support the evolutionists. But at this point, it's looking kind of bleak. And okay, you can ask the question, where did all the organisms come from? But here's another question. Is it really more rational to believe that, like I said earlier, just these molecules and atoms blowing around in space mindlessly, blindly, just kind of happened to figure all this out to get the right proteins and the right RNAs and the right DNA for the right circuits, for the right networks to create the right creature at the right time in the right place? With all these other, like, at any point, even if they did figure it out, they could have gotten, torn apart by a tornado or a lightning strike or something. See, to be an atheist, you really have to believe in a trillion miracles upon a trillion miracles happening for life to get. This is what people don't understand. They say atheists don't believe in miracles. Yeah, you actually have to believe in a lot of extremely unlikely things happening, because without God, you have no mind to guide the process. All you have is atoms blowing around in space. You're in a lot of trouble. You're basically saying, yes, I think that mindless, blind molecules somehow overcame miraculous odd after miraculous odd after astronomically unlikely event after astronomically unlikely event to produce life as we know it today in all its complexity and harmony and beauty. And there was no mind involved. That's silly. That's mythology. Really? Again, I think people should just open their minds to the various possibilities. I'm not saying anything is absolutely proven 100%. I'm just saying that there's questions to be considered, questions to be answered. And for the materialist, for the atheist, I think there are serious challenges and problems posed by all this. You really are relying on a system that is every bit as miraculous as the religious systems that are being rejected in their place. Okay, in my opinion. Again, someone else might rationalize all this and figure out a solution, but I would sincerely like to hear it. But yeah, so I'm going to end it there. For anyone who is willing to sit through all the boring words and scientific jargon, I think you really gained a lot from this. Because evolution is something that is taught and understood by many people to be, without question, an undoubtable theory. There's no scientific evidence which contradicts it, and that's constantly what the public is being fed. It's being fed in the public schools. They're being fed this in the universities. And it's just not true. It's just not true. And that's really important to understand, because without evolution, how do you get to where we are? That's just, I'm not saying it's God or intelligence. I'm just saying, what's the solution at that point? When evolution's gone and you can't get from one creature turning into another, where else do you go? What's the answer? You have to start thinking real hard to think of any. I've heard people say, "Well, we just don't know." Well, all you have is atoms moving around in space. That's all you have. Once that's been falsified, you have to go beyond atoms moving around in space. You have to go beyond materialism. In my opinion, again, I'd be willing to hear other people's opinions on this matter. I haven't heard anything convincing to me besides, if materialism fails, what's the other alternative besides intelligent design? But maybe someone else will think of something and put it forward to me. Anyways, okay. Thank you. That was ID update number 3. We will continue with these. We will continue to show massive problems with evolution, things where evolutionary scientists are teaching things incorrectly, kind of blowing up myths and things like that. There's so much information out there. A lot of it's boring, but again, it's fascinating for people who realize the implications of these things. All right, until next time. I don't see any questions, by the way. I don't know if anyone's even listening, but I didn't see any questions. So if someone had asked a question, I might have answered it, but I don't see anything. So maybe if you have a question, look in the put it in the comments. I don't know, but hope this was helpful. All right, until next time. God bless. Goodbye.